So in chapter 1, I told you at the beginning that it was a survey chapter. And in a survey chapter, you cover every topic that we're going to in this semester. So we started off with an introduction and said, what is biology? And we all came back and said, it is study of life. So we knew what biology was. Then we talked about the characteristics of all living organisms. Okay, we talked about that they need to be able to reproduce, that they had to have a chemical information system. And what's our chemical information? DNA. Fantastic. We talked about that they need to be able to evolve or participate in biological evolution. We talked about mutations and how mutations lead to adaptations, and adaptation leads to evolution. We said that there's nothing random in biology with two exceptions. And these two exceptions are mutations. Mutations are completely random, and the second one was crossing over, which takes place during the formation of gametes. And gametes, again, are sperm and egg. So in life, whether you are a human, a plant, a dog, or a fish, there will be no two sperm that are created that are the same, and no two egg. And because of that, it leads to variation. And variation leads to what? Survival of the fittest. And what does the survival of the fittest mean? When you say you're fit, what does that mean? You live long enough to reproduce. And how do I measure your fitness? How many babies you have. How many babies you have. You guys are fantastic. That's fantastic. Okay? So our keystone, we had two keystone points. The first was the two random things, which are mutations and crossing over. The second was that shape or structure determines function. Shape determines function. Structure determines function. If I change its shape, I change its function. I change its function. Right. So we want everything to keep its shape so it can carry out its specified function, so keeping it healthy. I talked about, and I used the example of the protein that was up there that kind of looked like squiggly lines that kindergartner drew. And I said, this is not random. And that's where we talked about the two things that are random. The example I used in class was the key. The key to my truck will start my truck, but it won't open my house because it has a specific shape, and that shape tells it to start the truck, not open the house. Okay? We talked about that. We talked about evolution, which we've already touched on, and the survival of the fittest. We talked about reproduction, mitosis, as far as cellular reproduction, and then, of course, sexual reproduction. You have to be able to grow and develop so that you can then reproduce. And then we got into classification, or taxonomy. And taxonomy is the science of classification. And we started with the most inclusive group, which was domain. And I said group, and let me correct myself, taxon. Taxon is a level within taxonomy. The most inclusive group, which means it includes the most number of organisms, is the domain. And then it gets more exclusive as it moves down. So we went from domain to kingdom, phylum, class, order, family, genus, species. We talked about the importance of genus and species in terms of binomial nomenclature, which is the two-part naming system, how we name or give scientific names to organisms. We talked about that in a scientific name, the genus and species are always italicized, and the genus is capitalized, the species is lowercase. We discussed that, and it made science a universal language, right? And we talked about applications for that. The one other thing that we covered that I missed first was the levels of organization. We talked about cells being the smallest unit of life, and then we integrated that into our emergent properties. And emergent property says we have cells, and cells do cells' jobs, but when we put a whole bunch of cells together, they work together to form a tissue, and then tissues to form an organ, organs to form an organ systems to form, and a bunch of the same organisms make a population. A species is a population, but it becomes more specific if it's in an area. So population, and a bunch of populations, community. We add non-living aspects, it becomes an ecosystem. Environmental aspects make it an ecosystem, and all the ecosystems form the... Yes! Yes, fantastic. That's what we did last time we were together, which was, I'm not sure which day, but it was that day. Okay? Now, we're continuing with taxonomy, but I wanted to be specific in how many domains we have and how many kingdoms we have, because you'll be asked those questions. So of all the living organisms on Earth, we can break them up into 3 domains. 3 domains. The domain bacteria, the domain archaea, and the domain eukarya. Anything that's alive on Earth is in one of these 3 domains. Anybody have an idea where you belong? Eukarya. This is you. This is your domain. This is your domain. Okay. What goes here? And what's the main difference? Single cell, fantastic. No nucleus. There is only one organism on Earth that is single-celled and has no nucleus. It's bacteria. Fantastic. It is bacteria. So I have 2 whole domains. I have 3 domains on Earth. And 2 of those 3 are composed of bacteria and 1 of eukaryotic organisms. What does this tell you about the number of species? Considering all living organisms, 3 domains is all they fit into. And 2 of those 3 are composed of prokaryotes. What does that tell you about the number of species on Earth? There are a lot of species, and there are far more bacteria than anything else. Anything else. So this is a whole domain of bacteria. Here's a second domain of bacteria, so things that we can't even see. And the third domain, eukarya, has anything on Earth that's alive and not a bacteria. So this is you, your dog, your fish, the tree, the grass, a bird, a snake, a sloth, a slug, an insect. Anything else that's alive that is not a bacteria falls into this kingdom. Does that make sense? No, I said kingdom. Domain. Sorry, I apologize for saying that. Sometimes I hear myself talk, and I'm like, did I say that? Call me on that. All right, I am going to come back to this slide in just a moment so that I can explain to you what this means, peptidoglycan. But I'll come back when we get there. So 3 domains. Now, domains are the most inclusive, and then we go down to kingdoms. Anyone have any idea how many kingdoms we recognize today in study? 6. Well, let me tell you where we started. When we first started classifying all living things, you were either a plant or an animal. If you're an animal, if you're not an animal, you must be a plant. Those were our only 2 options. We really didn't have much to go as far as microscopes were concerned, and we didn't know much about bacteria. We thought that all mushrooms and fungi were plants because they grew in the ground, so we were like, okay, they must be the same. So we had 2 kingdoms, plants and animals. Not too long ago, roughly the 70s, they realized there's more than just plants and animals. Okay? There's far more organisms than just plants and animals. So we had our plants and animals, but at that point it was clear that fungi were not plants. Does anyone know why fungi are not plants?
They don't use the sun.
They do not use the sun, so they do not carry out photosynthesis. Fungi eat their food. They have acid that they shoot out, and it breaks down everything around them, and they absorb their nutrients. They digest just like you and I do. Not as complex, but still. So that means fungi can't be plants. And are they animals? No. Because they have a cell wall, and animals are the only ones with no cell wall. So we had to make a new kingdom for just fungi. I get excited every time, sorry. Then we have the Protista kingdom, or protist. I'm a memorizer, so I have to always relate, like, how does this even matter to me? Protists or rejects? Rejects. Eukaryotic rejects. When you talk about an amoeba, that's a protist. A paramecium, that's a protist. Algae, a protist. And let me tell you why. Protists are eukaryotes that tried to be one of these, but didn't make it. But because they're a eukaryote, they can't be a bacteria, so we had to have a catch-all for the rejects. And guess what we called them? Protists. I feel like if you're going to have these mean cut-downs to your friends, like calling them a protist is pretty bad. Okay? If you tried to be my friend, but you're not. Okay? They're the rejects. So we have animal-like protists. Like an amoeba is an animal-like protist. It tried to be an animal, didn't make it, so we put it in that reject category. Okay? We have paramecium. There's tons of molds that tried to be fungi, didn't make it, so they're protists. And algae tried to be a plant. People still think algae is a plant, but it's not. It's a reject. So it's a protist. So that at this point we have these four kingdoms, and Monera was the kingdom of bacteria. So if you were a bacteria, we put you in the kingdom Monera. Now this is just 5. Today, we recognize 6, which we had already mentioned. So on this slide, I divided it to where I have the domain, because the 3 domains, and then I have the kingdom that's associated with it. So there was no confusion in your mind when you're studying. The domain bacteria has the kingdom bacteria. So it just so happens that they have the same name, and that works out well for you. The domain archaea has the kingdom archaea bacteria. So those 2 are kind of easy to memorize. The domain eukarya has the kingdom plant, animal, fungi, and protist. These 4 kingdoms fall into the domain eukarya. Okay? So today, you and I recognize 3 domains and 6 kingdoms. 3 domains and 6 kingdoms. Questions here? The next slide is a picture of this same information, but some people learn it better with pictures. Okay? So it's going to have the 3 domains and then the associated kingdoms that go with them. So we have the domain bacteria, kingdom bacteria. Domain archaea, kingdom archaea bacteria. The domain eukarya, kingdom protista, fungi, plants, and animals. Okay? So we recognize 3 domains and 6 kingdoms. Oh, and I put that here too. All right? Clarification needed right now. So when we divide you into your domains, whether you are bacteria, archaea, or eukaryotes, what is the defining characteristic of whether you're a bacteria or you go into the domain eukarya? What's the difference? If you have a nucleus or not. So I want to make sure you understand the difference between the 2. There are 2 options for cells. You can either be a prokaryotic cell or a eukaryotic cell. Let me clarify this, because a test question will say, what are the 2 types of cells? And some students write plant and animal. Plant and animal are 2 types of cells. You are correct. But when we are classifying organisms, there are 2 types of cells. Those with a nucleus, which are eukaryotic, and those without a nucleus, which are prokaryotic. So I want to make sure to put this out here and give specifics on this so that there's no confusion. The first type of cell is prokaryotic. When you do your word root next week, we'll start analyzing these terms. So pro means before, and kary means nucleus. So prokaryote means before a nucleus. So even if you didn't know what a prokaryote was, if you could dissect that word, it would help you kind of find that right answer, because it's a multiple choice test. Okay? And in high school we could say, you have a cell that has no nucleus. What is it? and we would say prokaryote. But in college, we don't say stuff that simple. We don't say, it has no nucleus, what is it? We say, this organism lacks membrane-bound organelles. That's the way we say it. I get that that's overkill, but I'm telling you this so that you recognize it when you see it on the test. We say, we have discovered an organism that lacks membrane-bound organelles. For example, a nucleus is a membrane-bound organelle. So if a cell is lacking that, what does lack mean? Does not have. So it's lacking these membranes, it must be a prokaryote. When I teach this to my high school freshmen, I didn't come up with this, one of my students did. She remembers pro, you do. Prokaryotic cells have no nucleus, eukaryotic cells do. So she said that one day, and I was like, you're a genius. Like, for people who memorize, that's perfect. Pro, you do. Pro, you do. So prokaryotic cells lack membrane-bound organelles. Look at the examples that I've provided here. What do they have in common? They're all bacteria. The only thing on earth that's prokaryotic is, and all bacteria are, there's no exception. None. None exception. All right, second type of cell is eukaryotic. Again, with our word roots, eu means good or true. Karyo means nucleus. So good or true nucleus. So a true nucleus is a eukaryote. Again, we don't just say, oh, this cell has a nucleus. We say this cell has membrane-bound organelles. Because we're intellects, and that's the way we speak. Examples. The only organisms that are eukaryotic are animals, plants, fungi, and protists. Okay? Pro, you do. Questions right here. Yes, sir.
So, what are the four kingdoms of eukaryote again? I'm missing one.
Animals, plants, fungi, and protists.
The fancy name for it.
Protista?
Okay.
For which fancy name?
I have
Go ahead.
Protista, Plantae, Animalia, and
Fungi. Yes, sir. Yes, sir. Yes, sir. Okay. Now, this is going to tie us back into the three domains in this next slide. Analyzing these prokaryotic examples. So everything on this slide, if it's prokaryotes, has to be what? Bacteria. And you can see that. Bacteria, bacteria, bacteria. But let's talk about the types of bacteria. The first group is eubacteria. What does eu mean again? True. So eubacteria is true bacteria, what you and I recognize as bacteria, and we can find it anywhere. The bacteria that's on you right now, on your teeth, on your desk, your pen, your clothes, that is eubacteria. Eubacteria. It's found almost anywhere. Does anyone know what cyano means, or cyan? Bluish. For those of you who go into anatomy or to a type of medicine, we actually use this as a diagnostic characteristic. If the patient comes in and they have like a bluish lips or their eyelids or their fingertips, we say that they're cyanotic. Cyan just means that they are blue, and usually they have that tint to them because of a lack of oxygen. So we use that as a diagnostic thing. So all across science, when I'm teaching you these word roots, they apply, not just to biology. So cyanobacteria is a bacteria that is bluish-green. Okay? So it's just a blue-green bacteria. This is a type of algae. Okay? And we'll get into algae and these small things later. The third example for bacteria is archaebacteria. I really want you to understand the purpose of archaebacteria and its classification because it's our whole domain of archaea. This type of bacteria is found only in harsh conditions, which is why I bolded that. Let me tell you where a harsh condition is. Inside a volcano. At the very bottom of the ocean, where there's those hydrothermal vents, which is a fancy way of saying underwater volcano. where it's super duper hot and there's a whole bunch of chemicals down there that are lethal. We find these bacteria. We find these bacteria in places where there's no oxygen, like the moon or the mud. There's no oxygen flow in the mud. We find these bacteria in areas like the Great Salt Lakes. Not like an ocean, where the salt concentration is high, but not lethal, but like the Great Salt Lakes, where if you were in the Great Salt Lake, for a short period of time, it would dehydrate you to a point of expiration. What does that mean?
Die.
You would die. I only find these bacteria in extreme environments because they need to be where nothing else can live. And I'm going to explain why. But they are only found in harsh environments. How many humans are you going to find in a volcano? Unless they're dead, right? You won't find any humans just hanging out at the bottom of the ocean. The pressure alone would kill us. Yeah, it would crush us. Okay? In the Great Salt Lakes we just talked about, we would expire. I'm going to go back so I can show you what the difference is, and then I'll come back to this slide. Right here. Regular bacteria, they have peptidoglycan. Archaebacteria have no peptidoglycan. Let's talk about peptidoglycan. Truly, all it is, it's a carbohydrate. But that may mean nothing to you right now, unless you're like, I'm on a low-carb diet, I don't want peptidoglycan, right? Let me tell you what it does. Peptidoglycan functions as like a protective mechanism. I explain it as being football pads. If I put my hand on this desk right now, which I already know is covered in bacteria, am I killing any bacteria? There's 150 pounds lying up on this desk right here. Am I killing any bacteria? No, because it's all protected in football pads, peptidoglycan. Now, if there were archaebacteria on this desk, first of all, they wouldn't be here. But if I leaned on them, would I kill them? The answer is yes. I would, because they have no protective mechanism. So why do they have to live in extreme environments?
Nothing else to bother.
Nothing there can kill them. They need to be where nothing can hurt them. They are very vulnerable, so evolution has allowed for them to have these random mutations that have increased their ability to survive in environments where nothing else can. Does that make sense? So now you know the difference between a regular bacteria, archaebacteria, which are only found in extreme conditions, and then everything else on Earth. We have 2 domains of bacteria and 1 of eukaryotic organisms. Regular, extreme, and everything else on Earth. Questions there? Everybody good with this slide? Okay? I wanted this picture for 2 reasons. First of all, to show you a diagram of prokaryote and eukaryote. Second, to tell you you don't have to be a genius to figure this out. Do you remember how we said prokaryotes lack membrane-bound organelles and eukaryotes have membrane-bound organelles? If I'm going to pretend I don't know what I'm looking at, so this is just what I would do. I would look at this and I would be like, okay, there's dark and light. I can see that. And if I needed to compare this one to this one, I can also see dark and light, but it looks like there's some designs in the light aspects of this one. Does that make sense? Those designs are held together by membranes. So the only way for me to have these designs is if there were membranes in it. And if there's membranes in it, it must be what type of cell? A eukaryotic. I don't have those designs when I'm comparing these 2. This is more specialized. This is still a protist. This is a reject. But this is a bacteria. Very simple. Not anything to it. Okay? So 2 reasons I wanted you to be able to kind of recognize that when you have all this, if it's in college and you have a diagram, there's a reason. Like we're trying to show you something with it. So I wanted to teach you kind of how to start easily navigating yourself with these diagrams. Questions on pro versus eukaryotic cells? Okay? Okay, last topic of chapter 1 is the type of research, biological research. So we're going to go through, there are 7 things to cover, and I've made a slide for each one, so this kind of just intros each one. All right, the very first thing is the scientific method. You and I covered the scientific method last week, and I actually asked you to do a write-up and let you pick whatever you wanted to write yours up on. And I didn't put any restrictions other than it can't be illegal and, like no one can die or anything like that. But a lot of you made up some really funny ones, and some of you were really straight to the point, which I'm cool with as well. The reality is there was 2 reasons for that assignment. The first was to get you to understand how to format a lab. Okay, and this is what's expected, to have you practice that. The second was to show you that basically anything you do can be tied back to the scientific method. And we talked about why you brush your teeth. We talked about why we wipe our butts. Okay? Let's not even get that scientific. What do I wear today? Am I working? Is no one going to see me? Can I just stay home all day? Do I have to go to the grocery store where somebody may or may not see me? Am I going out with my husband? Are we going to a volleyball game? What is it that we're doing? Based on what we're doing, I will pick my outfit. If I'm going to go watch volleyball, I'm probably going to wear long pants and long sleeves because those gyms are always really cold. Okay? If I'm going to the grocery store, I'm probably going to wear athletic shoes, athletic shorts, a t-shirt, and my hair in a ponytail because I'm trying to run in there, run out, and not let anybody talk to me. Okay? If I'm coming to work, I'm going to try to throw myself together a little bit. Okay? And if we're going out, I'm going to look cute. That's it. But I have a question, problem. I have different hypotheses depending on what's picking your outfit requires a scientific method. Even if you're like, I don't even care what I wear. That's still an observation. All right? So everything that we do, you wake up in the morning, my stomach hurts. What are your hypotheses? I need to poop. I need to eat. I'm hungry. Maybe I have a stomach virus. That's a thing. That's a real thing. Can we test it? Yeah. If I poop and I feel better, that was my experiment. My data says, based on my experiment, I feel better. My results show, my conclusion is, that the only reason my stomach hurt was because I needed to poop. That's a scientific method. So we did that last week. Just a quick on that. I'm not going to go into every single one of those. I think you get what I'm trying to say. Basic versus applied research. You guys are the kings and queens of basic research. You do this all the time. If I asked you, how many miles is it from here to the moon? What would you do? I would Google it. I would search it. Okay, in the old days, when we couldn't ask Siri or we couldn't ask any electronic device, what would we do? We'd go to the library. We'd pull up an encyclopedia. We would, some of you are like, where's the library right now? Because it's just not a thing. Because we have it all right here at our fingertips. But that's basic research. How many miles from here to the moon? Basic research. Applied research is when I take that information and I use it to solve a problem or answer, I don't want to say just a question. I'm going to say solve a problem. Solving a problem is answering a really intense question. So let me give you the same example. How many miles is it from here to the moon? Let's say they say it's 100 million miles. Siri tells me that. 100 million miles. Okay, now to apply that, I need to calculate how much fuel it's going to take my rocket ship to get from Earth to the moon. That's applying that information. Basic research is what I can quickly pull up. When you're with your friends and somebody brings up a new term, I did this in high school all the time. You're probably about to make fun of me. But in the spring, there was this girl. She was a mess. But she kept saying, you don't want no smoke to this boy. And I was like, of course he doesn't, because we don't smoke in the school. But I never get into their conversation. I've taught high school for 15 years. I can hear conversations going on. When you're supposed to be doing labs, and you're talking about who you hooked up with, I hear that. And I'm like, they should have done that. But I keep it to myself. But she kept saying that. And I couldn't, and she was a mess. Absolutely, but she was a mess. I couldn't figure out what she was saying. So I Googled it. I was like, What does that mean? Oh, he doesn't want any drama. He doesn't want any fighting. He doesn't want that. Okay, I get it. Now I understand their conversation. That's what I do. It's basic research. I figure out what it is that you're talking about, and then when I hear my kids say it, I already know, and they're like, Mom, you're not cool. I'm not trying to be cool. I'm just saying I know what you're saying. So quit trying to act like you're talking above my head. Okay, because I already know. All right? So basic research. I'm just trying to Google something real quick. Apply. You also do this, like, for example, Friday we have a volleyball game in Educator. I've heard of Educator before. I had no idea where Educator was. So what did I do? I did. I actually got through that maps, and I put in Educator. And they said, did you mean Decatur High School? And I was like, how do you know my life? Yes, weird, but you do. I need to go to Decatur High School because my daughter's in high school. She'll be playing at Decatur High School. It's 1 hour and 24 minutes. That is basic research. I know. Who does? Anyways, now I know it's 1 hour and 24 minutes. That's basic research. How do I change that to applied research? How much gas? Give me something else. After I ask it how far it is, when do I need to leave? And now our phones calculate that too. It also gives me different routes. Do I want to pay tolls? Do I not want to pay tolls? Yeah, you can do, that's applied research when you start putting it. So did I have to be in a lab coat or in a science lab to do all this? No. Okay? No. Data. 2 types of data. There's actually tons of data, but we're going to keep it simple. 2 types of data. Observational. Tell me what you're thinking with observational. You observe something. I see that there are 5 butterflies on this tree, and there are no butterflies on this tree. Butterflies like this tree. Did I do an experiment? No, I'm just observing. I see that this male lion is more attracted to this female lion than he is this female lion. Those are observations. Experimental is when I am actually conducting an experiment. Do you have to be in a lab coat and in a lab to do an experiment? No, we talked about last week. I want to know if people in Texas wave more than people in Virginia. My experiment is I'm going to wave at people in Texas and see how many people wave back. 5 people wave back in Texas. That's data. That's experimental. When I went to Virginia, only 2 people waved. That's data. That's experimental. I actually conducted an experiment. Okay? So observational and experimental. And we're all tying this to biology right now. So everything that I'm working with is living, right? Living. A hypothesis. I feel like we don't need to spend a bunch of time on this, but I did put here that, like I mentioned last week, testable and measurable. Hypothesis should be testable and measurable. We need to get away from saying a hypothesis is an educated guess, because there was some confusion on what we said educated means. Do you have to have a degree to come up with a hypothesis? No. What's an example of being educated? Just thinking?
Just being familiar with my topic.
Being familiar with the topic. Does experience count as education? Yes. Experience counts as education. My dad dropped out in 6th grade and runs a very successful business. Really great with numbers and cash. Dude is on it. Cannot spell for anything. He still puts thank you for your business at the bottom of, Daddy, send me your invoices. Let me fix those before you hit send. Okay, which he actually doesn't even email. He still writes out his invoices. And so I take them and type them and send them out. But he's very successful. Can he make hypotheses based on his experience in education? Yes. Okay, if a 2-year-old puts their finger in the socket and gets shocked, does that count as experience? Education. Yes. So we just can't say any further. Hypothesis is an educated guess. Hypothesis is a statement that is testable and measurable. And based, if you want to put education in there, but it's based on education/experience. Okay? So hypothesis we really got into last week. Here's a term that you definitely need to be familiar with. A null hypothesis. When I was taught this, it was so confusing because my professor was so incredibly smart and they were prided. This individual was very prideful of the fact that he was so intelligent. I never understood what a null hypothesis was until I watched it on YouTube. A null hypothesis is literally just the opposite of your hypothesis. But why couldn't Dr. Peck say that? I don't know. So if my hypothesis is that people in Texas will wave more than people in Virginia, guess what my null hypothesis is? People in Texas do not wave more than people in Virginia. So whatever my hypothesis is, it is saying that it is not true. You can't switch Virginia and Texas. You have to keep the statement the same, but all you add is the people in Texas will not waive. All I did was add not. Kept everything the same, so your null hypothesis is whatever your hypothesis is, it's the opposite. Whatever you think it'll do, it's what it won't do. In this class, you will not have to write a null hypothesis for grade. But in statistics, in any higher science class, you will. Okay? Right now, if your hypothesis is not correct, you can write in your conclusion, my hypothesis was not correct. In a statistics class, you would have to say, let's say your hypothesis is right, this is how you would word it. I failed to accept my null hypothesis. Why can't we just say, I got it right? I fail to accept means I don't accept my null hypothesis. And my null hypothesis was the opposite of my hypothesis. When you get into statistics and stuff like that and you hear that and you get confused, Khan Academy, he does it great. A little 10-minute video, null versus regular hypothesis, or pull this video up that we're recording right now to remind yourself. You need to be able to define this for your test and explain it, like understand it for your test, but you will not have to write a null hypothesis. Experiment. In an experiment, we're going to do lots of experiments. We designed an experiment last week as part of your lab, so I feel like you're familiar with experiments. The two things I want to point out here are that in most experiments, especially biological experiments, we have a control. A control means I already know what's going to happen, or this is what happens when nothing happens. So a control is I already know what's going to happen, or this is what happens when nothing happens. Let me give you an example. You will do a lab in here in 2 weeks where you will take a whole bunch of different foods in test tubes. It's not this many foods. It's like, anyways, you have a whole bunch of test tubes. And you're going to test, one of those tests is testing those foods for protein. And if you use the dye that I use and you put it in all your foods, the foods that have protein will turn purple. But before I have you put it with protein, I have you put it in water. Why do I have you put it in water? It's the control. When I add this indicator that only responds to protein, what's it going to do in water? Nothing, because there's no protein in water. So this is what happens when nothing happens. So I already know that. So when it does change colors, then I'm like, oh, something's happening here. Okay? So control. The second aspect of this is that in any hypothesis, in any experiment, what's the acceptable number of replications? How many times should you repeat it? 3. 3 is our basic, no, 3. You can't go wrong. Textbook, basic, 3 times you repeat it. Today in lab, and actually I can do this right now, I'm going to remind you that you need to know how many drops of water it takes to make 1 milliliter in a graduated cylinder. You have to know that. Okay? You have to know that. And I'm going to ask you to do it 3 times. Because if you got 15 drops one time and 22 the second time, at that point you're confused. So if you do it a 3rd time, you might have a better range. Okay? I also need you to understand, because I experienced this yesterday, there's a difference between a drop of water and a squirt of water. One student in a lab group, she was squirting it. 1, 2, 3, 5. And her partner said, no, it's just drops. And she was like, no, it's not. And so what do you think he did? He did it on his own. Like, he went and did it, like, on his own. Because he was like, I think this is what... And she was set in her ways, and she didn't ask for direction, so I just let her roll. She'll answer 3 squirts and get it wrong. Okay? But we don't know how many drops, and so I'm going to ask you to repeat it so that you have a good idea. That way, when you see the answer up there, you're like, okay, my number fell in this range. I know that has to be the right answer. So 3 times for as far as replicating, and we always set up a control. In this experiment, this one is straight out of your book, which is why I'm highlighting it. This is me, and this is my neighbor in the apartment next door. I put a plant, we both have plants, but I put a plant, and I didn't add fertilizer. In fact, I don't even have real plants in my house because they will die. And my kids are only alive because they talk. You think I'm kidding? Like, my husband's at the fire station today. Before we left, my oldest daughter said, what's for dinner? And I was like, she said, is Daddy home? She goes, so... I was like, oh. She goes, I'll make ramen noodles. I said, yes. Okay, perfect. It's thank goodness they speak. Okay? So, all my plants are fake in my house, though, when you come in my house. Not live, look. So here I am with my plant with no fertilizer, because it didn't tell me it needed fertilizer. And here's the lady next door who's got the same plant, but she fertilizes it. And her plant has a flower, but mine doesn't. So what do you think my hypothesis is?
Fertilizer.
Yeah. I bet if I add fertilizer, a flower will show up. Can I test that? Is it measurable? Yes. So here is my experiment. I took 6 plants of the same plant, and I didn't put any fertilizer, and I left them on my windowsill because I knew that I would be the one with no fertilizer. My neighbor has 6 of the same plant. Notice how they're all the same, not just random plants. All the same plant, and she added fertilizer to all 6 of hers. All right, so that's our experiment. There are 2 ways that this experiment could end. The first way, my plants didn't get flowers, and her plants didn't get flowers. So what do you think I would conclude? Yeah, adding fertilizer doesn't control whether or not a flower shows up. That's a good conclusion. Another way that this experiment could end, mine could still have no flowers, surprise, but hers did have flowers. And my conclusion would reflect what? In conclusion, based on the data, adding fertilizer does stimulate the growth of flowers. Okay? So you have your experiment, you have your control. There are different ways that it could end up. Just because my hypothesis is wrong, do I say, do I change my hypothesis? No, I just say, hey, look, fertilizer doesn't cause flowers to grow. All right? So this is an example of experimental research straight out of your book. Questions here? Okay, model organisms. This is not brought up on the notes on eCampus because it's just like mentioned, but I need you to know how important model organisms are. So on the new version, I dedicated a whole slide to model organisms. Normally when I teach this topic, I say, does anyone know what a model organism is? And they're like, it's a good-looking organism. And that may or may not be true, but there are 4 criteria for a model organism. When we say a model organism, we're talking about this is an ideal organism to test on. This is a model organism for experimentation. This is a model organism for experimentation. The first criteria to be a model organism is that you have to be able to reproduce quickly. Guess what humans cannot do? Reproduce quickly. They can become pregnant, and then 8 to 10 months later, they'll have the child, and usually it's just one. Of course, it could be multiples in some cases. And then you have to wait 18 years for that kid to mature by, society's guidelines, 18. We don't reproduce quickly. So if you did a test on me and wanted to know how it impacts my child, you would have to wait a long time. What's the type of organism that reproduces really quickly? Rabbits, rats, or mice. Okay, plants, bacteria. So what do we do a lot of testing on? Rabbits, rats, mice, yes. Second criteria to be a model organism, you have to be easy to maintain. I don't care if you say, I'm low maintenance. You are not low maintenance. Okay? You are a high maintenance person. But bacteria, if I want to feed bacteria, I can get a can of chicken broth for like 50 cents or $1 at the grocery store. I can pour it into their little trays. I can feed billions and trillions of bacteria for 3 or 4 weeks on $1 can of chicken broth. That's easy to maintain. And when I'm done with those bacteria, I can drop bleach on them and kill them. Do I get in trouble for that? But if I just fed a human a can of chicken broth for 4 weeks, and then I was done with them, gave them bleach, would there be some consequences? Potentially. Okay? All model organisms have to have genetic material. What is that? DNA. We use DNA in all living organisms, and let me give you an example. Those people who are diabetic are only able to have the amount of insulin that we have now because of bacteria. We took the gene in a human that makes insulin, and we put it into a bacteria, turned it on, and got the bacteria to make insulin. Then harvested that insulin from the bacteria, put it into those vials, and sent it off and sell it. We were doing it with animals, but large animals didn't reproduce fast enough. How fast can a bacteria reproduce?
Seconds.
Usually like an hour or so, quickest one. Long term, 24 hours. Bacteria are having babies about that. But that's because they clone themselves. It's not sexual reproduction. Clone. The last characteristic of a model organism is no one's upset if they die. So when I don't water my plants, no one comes after me. Oh my gosh, did you kill these plants? Yes, I did. And I didn't water them. Did those bacteria die in your lab? Probably so. Nobody cares. So model organisms are model because they're ideal for testing purposes. So when you hear the term model organism in a scientific reference, it's because they're ideal or model organisms for testing. And these are those criteria to become a model organism. So like you guys said, rabbits, mice, bacteria, plants, animals do not fit into this. Like large animals, like you and I. And if you are part of a test or an experiment, what have you signed in advance? A waiver, because it is illegal to test on humans. So if you have cancer, terminal cancer, what's terminal mean? You're on your way out. And you have a research group that comes up to you and says, we have this cure for your terminal cancer. But it's not been approved by the FDA yet. Do you want to try it? And what do most of those terminal patients say? Yes. Because what do they want? To live a little bit longer. I'll pay you $60 a day to take my medication, but you have to sign this waiver that says, if you die, I'm not responsible. And I don't have to keep paying you after you die. Do a lot of people sign up for that? Yeah, they do. A lot of people do. Especially people who don't want to work but want money. They'll take whatever, put it in their body, and get paid $60 a day and be good. And then those medical companies can collect data, show it to the FDA, and say, look, I gave this guy my medicine, and he lived. He was given 6 months to live, and I let him stay alive for 9. It's because of my medication. And if I can get that to happen enough, they'll say that my medication is okay, and then I can start selling it. But it's not okay to just say, hey, this class, I'm going to try this medication. You can't do it. You can't test on humans. Nope. Theory versus law. In biology, we reference theories. In physical science, we say laws. What's the difference between biology and physical science? Life. And there's always a chance in life that things could change or go wrong. Okay? When you're talking about physical things, like gravity, when you calculate the speed of gravity, 9.8 meters per second squared. Like, that is a solid. What's the number for pi in math? 3. That's you know what it is. It's not going to change. The cell theory says that the cell is the basic unit of life. And based on our research right now, the cell is the basic unit of life. But is there a chance in the future we might find something else? Possibly. So can we call it the cell law? No. Okay? If there's a chance that something could change, we consider it a theory. Is there a lot of evidence that tells us the cell is the basic unit of life? Yes. There's a small chance it could change. Okay, so theory versus law. Laws cannot be disproven under natural circumstances. So the laws of thermodynamics, anything like that, Newton's law, all of those are solid. Theory usually has something to do with something that could potentially change. The example that is referenced in your book is Alexander Fleming's discovery of penicillin, which he did by accident. He was testing bacteria. This gentleman was a bacteriologist. What does he do? Study bacteria. Ology is the study of, and then bacteria. So bacteriologist is an individual who studies bacteria. He set out a whole bunch of bacteria and grew them in petri dishes. And whenever you are growing bacteria in petri dishes, you should cover them. And most of you who have been in micro or anything like that, or even in your high school biology class may have done that, you cover it. So you let it grow, that way it's not exposed to anything. Well, he recorded all his information and accidentally left out that on one round he left them open. So he exposed them to everything else in the air. Then he came back and covered them, but a little while later he realized there was a mold growing on these bacteria. And that mold was actually killing the bacteria. And he was like, awesome. I found something that will kill bacteria. This is a type of bacteria that harms humans. So if I can get this mold and put it in a medicine, I can give it to humans and it will kill those bacteria. That's what an antibiotic does. He did it one time, but when he tried to replicate it, guess what he couldn't do? Couldn't replicate it because it was just a random thing that he had left it uncovered. Like he didn't think that that was like part of it. But he recorded all his information, and almost 100 years later, people replicated what he did because he kept such great notes and were able to create that same mold, which they isolated as being penicillium. And penicillium was used to create penicillin. And penicillin is the #1 prescribed antibiotic now, unless you're allergic to it, and then it's a mox. But penicillin is an antibiotic. Anti-bio. What is bio? So it kills life. Antibiotics will kill bacteria in your body if you have a bacterial infection. But what if you have a viral infection? You can't kill it. Why can't you kill it? It's not alive. It's never been alive. So if you have a virus, you go to the doctor, got a horrible stomach virus, what's the doctor going to say? You're just going to have to poop that one out. There's not anything I can give you that will kill that virus because that virus is not alive. I can treat your symptoms. Are you nauseated? I'll give you medicine for nausea, but I cannot get rid of the virus. Okay? This is the end of chapter 1. Any questions or clarification needed here? Okay, let's talk about real quick your lab.